guys, Sarah here. So today I'm going to be going over how to calculate two different loci with three different gene mutations, two of those gene mutations being allelic to each other. In the previous video, we calculated double mutations without any of the allelic stuff going on. So if you want to get comfortable with doing double mutations, I highly recommend watching that video first. That's going to be probably the easiest video to follow along before you move on to this. This is a little bit more complex. And there's also going to be a lot of other complex stuff in here as far as Punnett squares go. So if you're not comfortable with Punnett squares yet, you definitely want to go back and watch the first video in this series. I will link both of those videos in cards above here in all of this, so you guys can easily go link to that and watch them first. I also would really appreciate it if those of you who are watching who have not subscribed, go ahead and subscribe. I do these genetics videos pretty much weekly, and there's always going to be more genetics videos to be done. So if you like these genetic videos and you learn a lot from them, please subscribe and share, and comment below and let me know if there's any specific one that you'd like me to go over. Now what we're doing today is calculating, as I mentioned, three different genes but on two different loci. And if you'll remember from previous videos, a locus is a place on the DNA where the information for the gene is placed, and an allele is what holds the information for that gene. And a lot of genes in corn snakes are allelic to each other. I actually have those here for you guys. A lot of this stuff was on the board before. I didn't change a whole lot, but I do have the dominant genes, incomplete dominant genes, and the recessive genes like all others. But I also wanted to mention these that are recessive and also incomplete dominant with each other, or allelic to each other. So there's those. I didn't put Motley and Stripe on here. I'll do Motley and Stripe again in a future video as far as because the visuals are different, since these are incomplete dominant, and they are allelic just like Motley and Stripe are allelic, but these will actually change the visual phenotype of the animals when they're combined, whereas Motley and Stripe does not always change the phenotype. So I didn't want to put those on there right now. We might do a video on that in the future. So I went ahead and put the hypothetical pairing up here, and if you'll notice, I have our typical J and V for genes 1 and 2, and I went ahead and added the K for gene 3, which we're going to hypothetically say is allelic to the J gene. And then X, of course, is our placeholder, as I've done in all of the previous videos. And if you'll remember, you have to go through when you're making this Punnett square and associate each side, each possible way that the two different sides of the DNA could line up with each other. The first one is going to look very similar to the last one, where we had a lot of J's and V's lined up together. So if you'll remember, this J here could tag up with this V or this V. So we're going to say JV and then JV again. And then the second J could tag up with this V or this V also. So then we have JV again and JV again. And over here is where it gets a little bit more complicated, since we have other things going on. So this J could go with V or X. So we're going to have a JV here, JV, but then we're going to have a JX. And then this K could also go with the V or the X. So we're going to have a K, X, and a KB. I kind of did that in the wrong order. Here, I'll put it in the right order for you guys. There we go. So this is how we lay out our Punnett square. And I'm going to go ahead and make the square. The reason I am actually making the squares in front of you guys this time, as I mentioned in the last video, is because I know a lot of you are probably following along with a pen and paper, and it'll be a lot easier for you to do this with me than to have to pause the video and continue to catch up. So I will go ahead and finish this square off. So remember, we want to separate each of the pairs like so. I'm going to do it this way. There we go. Okay, and if you're confused by any of this, I highly recommend checking the last video, but I'll kind of briefly go over it again. So this double J means that the animal we're talking about is homozygous J. I like to put any homozygous genes together next to each other, and I think a lot of other people do that too. But this means, so this is one side of parent 1's DNA, and this is the other side. And then this is one side, and this is the other side. And then this over here, being parent 2, is the first side and then the other side, and then the first side and the other side again. Now, in this pairing and in most other pairings, it doesn't really matter which gene is on which side. There's still an even possibility of everything lining up in the way that we've seen here. So in this case here, we're working with 4 different letters, and so that makes it easy for us to see that this J and V can be together, J and X can be together, K and V can be together, and K and X can be together. And there's a 1 out of 4 chance that that will happen in any situation. Now that does change with a couple of genes in corn snakes, but we're not going to go over that right now. We'll go over that later on. Because I know the J and V gets confusing, but that is why these line up this way. It's because each of these stand for a different side of the DNA. I know that in the last video I put a dash between these so that you guys could kind of tell where the pairs section off, and that is something that I do to make it easier for me. If you guys would also like to do that for yourselves, feel free to do that. And again, I will also mention that if you guys would rather use a small letter in place of the X, feel free to do that. I use X as a placeholder, but most people, if you go to a biology class, instead of saying VX, they'll have you use capital V and small v. I don't like to use capital V and small v or that with any letters when I'm doing this, just because it makes it confusing since the letters look so similar, and I'm not in a place where I have lined paper and it's easy to show which one is the larger letter and which one is the smaller one. So that's why I use X as the placeholder. It's up to you, whatever you want your placeholder to be. Just keep it in your mind, whatever that is. Okay, so I'm going to go ahead and go throughout our Punnett square and fill this out. You guys may have done this already if you're following along, but we're going to have JV, JV all along here because this JV is going to go all the way across, and then each of these JVs as they go down are also going to fill in. So I'm just going to go ahead and fill in the first row completely since we already know that that's what it's going to be. I'm sorry if I'm blocking anybody's view here. Just need to get all this filled in. JV. Okay, so I went ahead and filled that in, and I'm going to go ahead and fill in all of these JVs downward. Since we already know that the second half of each of these is all going to be JV, I'm going to go ahead and do that as well. And remember, like I said, as long as these pairs stay in pairs, it doesn't really matter for now what order they're in. So if you have VJ instead of JV, that's okay. As long as you can keep track of which is which, that's all that really matters. Okay, so all of our JVs from the top are put into their spaces down below. So it's time to go ahead and move everything else across. Now this is JX, which means more of the heterozygous J. So I'm going to go ahead and put JX all through here. JX, JX. And then this is KV, so we're going to put K and V. KV. And then this is KX, so we're going to do K and X. And I'm sorry that I said this was heterozygous J. This X actually comes from the vacant one on the V locus. So KX, KX, KX. Okay, so now that the Punnett square is all filled out, it's time to actually move these over here and apply them to other genes one at a time. So what we're going to do here, everything over here is JV, JV. So I'm going to go ahead and pair up the J's with each other and pair up the V's with each other because that's what the genes that actually line up with each other. So all of these, 1/4 of all of these are going to be JJ, VV, JJ, VV, which is still the same as JV, JV. It's just, like I said, I like to put the pairs together when they do line up so that I can just glance at it and say, oh yeah, it's homozygous J, without having to sift through everything to find those letters, figure out what we have. Again, here we have the same thing going all the way across. So we have JX, JV going all the way across. And if we put our J's together, so this is 1/4, we can put both of our J's together, and then everything else is going to be XV, which is the same as this VX. I know that like I said, they're switched around, but in this case, it doesn't really matter that they're switched around. We just know that that's heterozygous V, homozygous J. These are where it gets a little bit different. So we have KV, JV. And so what we're going to have here is homozygous V, and then heterozygous K, heterozygous J. And remember, since J and K are allelic to each other, meaning they share a locus, they can appear next to each other just like they do up here. So we're going to, we also have 1/4. The reason I'm saying 1/4, if you didn't notice, is because this all is 1/4 and it's all the same. This all is 1/4 of the total and they're all the same. So each row here, they're all going to be the same. That will be different in future videos, but for now, we all have this 1/4 breakdown. So we have 1/4 VV, and then we have K and J. So remember, like I said, this K and J can be next to each other because they're allelic. But if they were not allelic, they wouldn't be next to each other. We'll go through that in a future video as well, don't worry. I know there's a lot of future videos, and like I said, if you haven't subscribed and you like this kind of stuff, there are a lot of future videos to come, and they're all really interesting. So here we have the last one, 1/4, we have our KXJV. And this is the hardest one because we have all 4 letters all in one space. But if you remember that when the K and J are together, they're allelic, so they can be paired up. So all of this is KJ and then XV, because that X is the vacant X up here next to the V. So what we have here, these are going to be the results of when you have, this is a pairing that's going to be similar to like a snow with an ultramel het anery, something like that. So if you have a pairing like that, these are going to be your results. And like I said, we will apply all of these to different genes later, but this is kind of the type of thing that it would show up in. So what we have here are 1/4 JJVV, 1/4 JJXV, 1/4 VVKJ, and 1/4 KJXV. I'm sorry if I was saying G instead of J. It's just a slip of the tongue there. So why don't we go ahead and apply things to these, apply different genes to these, just to sort of give you guys an example. I already mentioned the ultramel and the anery one, so we'll just do that first. So let's assume that the V is anery, and the J is amel, and then the K is ultramel. If we apply this to all of these, we will end up having this 1/4 snow, 1/4 amel, that's het for anery, 1/4 anery, that is also an ultramel. Sorry, it took me a second to read that. So 1/4 ultramel anery, and 1/4 ultramel that is het for anery. And like I said, I know this is kind of confusing because we're talking about 3 different genes, but if we just, here, I will go ahead and put this into ultramel and anery terms. So our J's are A's, so this would be an A, N, N, which is snow. And then if our J's here are also amels, and then we'll have a het anery, so that's amel het anery, and then 1/4 anery ultramel, those are backwards, but that's what that is. And then 1/4 we have ultramel het anery. So if that makes a little more sense to you, and if you do, like if you go to say like morphmarket.com and you put in these parents being a snow and an ultramel het anery, these are going to be the exact same things that it will pop out as the possible outcomes for that pairing. And we can apply this to a few different things. So we could, for example, instead of amel and ultra, this could be like hypo and strawberry. And then instead of anery, we could say diffused. And you can play around with all of this stuff, figuring out what's what. The only times that it's really going to get super confusing is when you start talking about your dominant or incomplete dominant genes, the incomplete dominant that are to the normal type instead of with each other. That's where it gets kind of confusing, because then you have to really look at, okay, palmetto, for example. If the V is palmetto, then that changes the way the snake looks, as opposed to another recessive gene where you would have like normal's het for something or ultramel's het for something. Like this would be an ultramel het palmetto in that case, and it would be a lot lighter than a normal ultramel because palmetto is kind of a hypomelanistic type in heterozygous form. And then when you have your dominant genes, if this V was a tessera instead, you'd have to remember that all offspring would be visual tessera along with the other genes that you're calculating. So you can play around with this. If you guys have been following along in this series of videos, you're smart enough by now to know like how to put the genes that you are doing into something like this and interpret it yourself. If you have not figured that out yet, that's okay. This is a lot to take in, and honestly, I'm still learning this stuff, and it's only going to get more complex. So if you still have questions, it's okay. Go ahead and leave them in the comments. I'll try to answer them. Either I will comment back to you, or I will also address it in another video. Normally, if I do answer a question in the comments, I will also answer it in a future video, especially for these genetics things, because they are really complicated. I really do hope that this helped you, though. I hope you kind of understand how to put things together. This wasn't a whole lot different from my last video, but it was different enough in that we did add another gene so that you guys could sort of learn how to interpret, how to calculate an allelic gene into your double loci calculations. So thank you for watching. Let me know in the comments below, like I said, if you had any questions. Subscribe, share this video if it helped you, and I will see you in the next one.